know that the advanced epidemiology study can enhance the knowledge how we understand the transmission of the diseases. And then for this event, we invite Professor Paul from Malaysia to talk about the his perspective, his knowledge, his expertise in Plasmodium knowledii as a zoonotic malaria. Using the molecular study, we will hear a lot about the his experiences, his knowledge, and his insights about the understandings, the complexity of transmissions of the zoonotic malaria, Plasmodium knowledii in Malaysia. And then he is also the expert for the population genetics, how we can understand about the genetic study in the community, in the public health, and then we can ask the questions to Dr. Paul. And I encourage to all the students from the Master of Epidemiology and from the Faculty of Public Health or outside of the Faculty of Public Health to ask the questions to Prof. Paul. He is the expert. Okay, and it's very rare event. We invite him, and then we can ask many ideas about the population genetics, about the modern epidemiology, about the molecular epidemiology, how we can understand about the transmissions of the zoonotic malaria or other communicable diseases. And once again, thank you very much, Paul, for joining us today. Even though this is the Saturday morning, and then he agreed to talk about the this topic to our students. Thank you very, very much, Paul, for coming and for joining us this morning. And I really hope that maybe in the coming months, we will try to invite Paul, Prof. Paul, to come to Semarang, and then we can talk deeply about the collaborations and about the topics of the research or even the academic exchange. We have many programs from the university. Prof. Morsid will talk about it, and then maybe we can strengthen the collaborations in real time, and then we invite you to come to Semarang and then talk about it. And once again, for the participants, for the students, it is a good opportunity for you to ask the questions to the experts. And I hope you really enjoy for Dr. Paul's speak today. And I hope we can get knowledge and we can get the insights from the modern epidemiology from Professor Paul. Once again, welcome, Professor Paul, and welcome everyone. Thank you very much.
Thank you very much, Dr. Fauzi, for the warm and inspiring words. We are truly appreciate your presence and support in today's online session. All right. For the next session, we will have the opening remarks from our Dean's representative because our Dean, Dr. Budiono, could not attend this session due to another agenda. So we are honored to invite Professor Dr. Mursid, Master of Science, on behalf of our faculty, staff, and participants joining us online. We extend our warmest welcome. To Professor Dr. Mursid, Master of Science, the screen is yours.
Thank you very much, Nabila. Assalamualaikum warahmatullahi wabarakatuh. Good morning, ladies and gentlemen. On behalf of the Faculty of Public Health, Universitas Diponegoro, I am really honored to welcome all of you to this important academic occasion. We are specially dedicated to host Professor Paul from Universitas Sarawak, Malaysia, as our visiting professor today. The theme of the lecture is Molecular Epidemiology of Zoonotic Malaria, Understanding of the Complexity of Transmission. Is highly relevant for our faculty and our community. As we know, zoonotic malaria represents a significant challenge for both research and public health practices. Understanding its molecular epidemiology is essential not only for advancing science, but also for shaping policies intervention that protect population and endemic regions. We believe in this visiting professor program will not only enrich the knowledge of our students and faculty members, but also strengthen the collaboration between our institution and distinguishing researchers from around the world. Such collaboration is at the heart of our academic mission which is to integrate education, research, and community engagement. I would like to extend my sincere appreciation for Professor Powell for accepting our invitation and sharing his valuable experience with us today. I also thank the Master of Epidemiology program for organizing this event and all faculty members and students for their active participation. I agree with Dr. Fauzi. Next month, October or November, Dr. Fauzi, we will
As soon as
invite
possible, Prof.
Prof.
As soon as
Okay,
possible after Professor
yeah,
Paul available.
okay, yeah. I also concerned in malaria, but anomalous with the molecular study in East Kalimantan. So, my decision ensure all of our continuing striving on the excellence in the research, teaching and suffices to contemplate meaningful global. With the Bismillahirrahmanirrahim, this event I will open. Assalamualaikum warahmatullahi wabarakatuh. Back to Nabila.
Waalaikumsalam warahmatullahi wabarakatuh. Thank you very much, Professor Mursid, for official
Yeah.
opening today's event and for the continuous encouragement toward
Mm.
academic excellence and international collaboration. All right, everyone, we are getting close to the sharing session today, so I think it's a good time to make this event more memorable, right? So I kindly invite all of our honorable guests and participants to please turn on your camera for our first photo session. So, and if it is possible, please kindly use the virtual background that the committee has sent in the chat room. Our documentation team will assist in capturing the moment. So we will wait for maybe over 3 minutes for everyone prepared and turn on your camera.
Nabila, you stay muted.
All right. All right, let's bring out our brightest smiles. Are you ready, everyone? Okay. On the count of three, I will count. 3, 2, 1, smile. Okay. For the next slide. 3, 2, 1, smile. Okay, this is the last slide. Be prepared. 3, 2, 1, smile. Okay. Thank you everyone for joining this virtual photo session and for capturing such a beautiful moment with us. Okay, next, we would like to inform all of the participants to kindly fill out the attendance form by scanning the barcode on the screen or just type the link. Before we go to the main session, to the committee in charge, please share the link. Okay, so all the participants, you have 5 minutes to fill out the attendance form. Thank you. Okay, since everyone already filled the attendance form, we are moving into the highlight of today's event, the sharing session with our outstanding speaker, Professor Paul Cliff Simon Davis. But before we begin, it is our pleasure to introduce our distinguished speaker, Associate Professor Paul Cliff Simon Davis from University Malaysia, Sarawak. So, to the committee in charge, please kindly share the CV. We are moving into the highlight of today's it is our pleasure to introduce our distinguished
He's
speaker,
currently the director
Associate
of the
Professor
Malaria
Paul
Research
Cliff
Center
Simon
and an
Davis from
associate
University
professor at the
Malaysia,
Faculty of
Sarawak.
Medicine and Health
So, to the
Sciences.
committee in
And
charge,
Professor Paul
please
earned
kindly
his
share the
PhD in
CV.
parasitology from the London School of Hygiene and Tropical Medicine after completing his master and bachelor degrees in Malaysia. And his research focuses
We are
on
moving into the
malaria,
highlight of
zoonotic
today.
disease, and public health. And he has led many important projects across Malaysia, Indonesia, and the region. He
It
is
is our
widely
pleasure
recognized
to
for his
introduce our
contributions.
distinguished
He's
speaker,
currently the director
Associate
of the
Professor
Malaria
Paul
Research
Cliff
Center
Simon
and an
Davis from
associations
University
to his
Malaysia,
publications,
Sarawak.
numerous international
So, to the
collaborations,
committee
and
in charge,
prestigious
please
awards,
kindly
including the
share the
PhD in
CV.
Silver
Silver
Medal
medal
in
in
Parasitology
London
and
School
the
of Hygiene
Gurnam
and Tropical
Prize
Medicine
for
after
Best
completing
PhD,
his master and
PhD
bachelor
thesis.
degrees in
Beyond
Malaysia.
research,
And
Professor
his
Paul
research
has been
focuses
active
on
on
into the
malaria,
highlight of
zoonotic
community health
disease, and
programs
public
and
health,
malaria
and he
awareness
has
campaigns.
led many
He has
important
also
projects
served as
across
an invited
Malaysia,
speaker
Indonesia,
in
and the region.
conferences across Asia
He
and
is
the our
widely
so, without further ado, I would like to invite Associate Professor Paul Cliff Simon Davis and everyone, please give your warm virtual welcome to the
collaborations
committee
and
in charge.
prestigious
Hello,
awards, including
Professor. If there is no delay, please join me.
Right. Is it my turn now? Sorry.
Yes, it is.
Okay. Sorry. Thank you very much for inviting. Actually, what I would like to say before I start, I would like to say great appreciations for Dr. Fauzi for actually inviting me, contact me personally to actually share my expertise, my knowledge on this very important topic. And also, I would like to thank Vice Dean for Prof. Mursid for welcoming me here as well. Thank you very much. Um, Although I haven't reached Undip yet, I really feel I'm already there right now. So hopefully I will reach there one day. Okay, so let me share my screen. Okay, I hope you are able to see my slides here. Right. So, malaria, I've been working on malaria, just a little bit of my background, working on malaria since I think almost 20 years already. So, particularly on the zoonotic malaria. So, therefore, today's topic is what we have been actually working on understand the transmissions of zoonotic malaria that is pretty alarming in Malaysia, particularly in Malaysia and Borneo. So before I start, I just want to introduce you about the Malaria Research Center, which was actually established as a center of excellence in the University of Malaysia Sarawak in 2006. And this center is located at the Faculty of Medicine and Health Sciences, where it was first founded by Professor Babir Singh and Professor Janet Cox Singh during that time, where they have a huge grant from the Wellcome Trust UK, and also where they have described the huge natural infections of Plasmodium nosai in Sarawak. So the MRC here, the main strength here is where we study the epidemiology, the clinical aspect, and the population genetics of the malaria parasites, not particularly on zoonotic malaria, but also on other plasmodium that are infecting human as well. So this is to show you the where Israel is located. So we share a very huge border with Kalimantan. Yeah. Okay. And just to show you where is the Kuching and the Kuching and Kota Semarang located, the university here and where I am. And Kapit is actually where is our study sites of zoonotic malaria. So basically, it's a very long journey from Kuching to Kapit, where Kapit is actually a very the largest divisions in Sarawak, where there's lots of zoonotic malaria infection there. So it's actually confined in that area compared to other regions in Sarawak state. All right, so the early landmark discovery of MRC is actually, like I mentioned just now, where the team has identified a large focus of plasmodium infections in humans in Sarawak. So this is actually the original team where I first started in the lab as a master students. I'm at the back there with brown shirt with hairs. And now you notice now I don't have hairs anymore. Okay, so and the other landmark discovery where we identified that wild macaques are the reservoir host of the infections, and also the team have described the clinical features of nosai infections, and also the treatment of management where it is actually where the WHO and the Ministry of Health are based on when it comes to treating zoonotic malaria. So the team also have incriminated Anopheles latent as the mosquito vector that transmits the parasites from the wild macaques to humans in the area. And we have been collaborating with lots of universities and agencies, not only in Malaysia, but outside Malaysia, particularly with the London School of Hygiene and Tropical Medicines, with the Western Australian University, the Ministry of Health, and so on. This is just a few that I listed out here. All right. Now, going back to the topic, the important topic here, just want to introduce you briefly about the infections of zoonotic malaria in Malaysia, particularly in Sarawak. Just for your information, that Malaysia is divided into East Malaysia and West Malaysia. So East Malaysia is where Sarawak state and the Sabah state are located. And actually, in Malaysia, the Sabah state have the highest zoonotic malaria infections, followed by Sarawak. Now here, I'm showing you about the case that has been reported from years to years. Now, if you look at this plot graph here, you can see that the nosai infection that is colored in black have been more than 70% every year of all the total cases, particularly in Sarawak. You can see that we also have human infections malaria by the human plasmodium species, but most of these are actually imported. So we do not have indigenous infections of Plasmodium falciparum or vivax and so on. So if you look at this on the right, the zoonotic malaria 2024 in 2004, in that year alone, the nosai infections cover 79% of the total malaria in Sarawak, and you can see that the hotspots of this zoonotic malaria are in Kapit division alone. Now, has been the high cases of nosai malaria in Kapit has been actually steady but high. Okay. Just a brief bring you back before we go to this population genetics of Plasmodium nosai. So this is actually the history of our the backbone of our studies, that where we identify the large focus of human Plasmodium nosai infections particularly in this capit division. So in early 2000s, there is no actually official report of Plasmodium nosai because all of these were actually identified by microscopy and reported as Plasmodium malariae. Now, it has been a bit, how to say, a bit bizarre that there were high cases of Plasmodium malaria in that area. So during that time, Prof. Babir Singh discussed this with the Ministry of Health to actually examine this particular Plasmodium, so-called Plasmodium malariae, and then design a molecular assay to identify the species of this so-called Plasmodium malariae. And using PCR methods and DNA sequencing of the small cell unit ribosomal RNA genes, that it is confirmed that these are not Plasmodium malariae, but instead they are Plasmodium nosai. So this is actually the history of how the large focus of human Plasmodium nosai infections were identified in Malaysia during that time. And this was published in the 2004 paper and has been continuously cited when most people, when they talk about the large focus of human Plasmodium infections in Malaysia. When we identify this nosai malaria in humans, so of course we need to know the source of the infections to understand how humans were actually infected by Plasmodium nosai, because we know that even in old publications, we know that Plasmodium nosai is actually a natural infections in monkey. So during that time, we also we caught wild macaques in capit divisions and examined them using a molecular method, a very simple PCR assays, and also DNA sequencing of the small cell unit ribosomal RNA genes to confirm the species. And we found that out of more than 100 monkeys that we caught in the forest of these capit divisions, almost all of them were positive for malaria parasites for Plasmodium species. And we see that they are infected with different species of Plasmodium that are commonly found in wild macaques, which were Plasmodium innoi, Plasmodium nosai, cornea, cynomolgi, and villi. And you can see here that majority of these monkeys were infected by innoi and nosai and cornea. Multiple species infections are very common among these wild macaques. It is well understood. It is as expected because they are in the wild and they're intense of there's forest mosquito in the wild. They're able to transmit the parasite from one macaque to another macaque. And we also caught two types of macaques here, long-tail macaque, Macaca fascicularis, and also pigtail macaques, the Macaca nemastrina, and both actually harbors Plasmodium nosai infections in the wild. Right. So to understand whether these macaques were the source or the reservoir of these nosai infections in human, so we further investigate using this genomic analysis of the mitochondrial genomes of Plasmodium nosai, both in humans and also in the macaques, and we determined whether or not these macaques are the reservoir host by looking at this haplotype network of the mitochondrial genomes um, of nosai from both from macaques and from humans. So interestingly, it is very important to understand the nature of these mitochondrial genomes to understand the infections here, because compared to the nuclear genomes, the mitochondrial genomes are maternally inherited, so therefore there is a lack of genetic recombinations. They are highly conserved. You can actually tells the history, the evolutionary history of the parasites, how they evolved in the macaques, and so how they actually transmitted to humans by just looking at this mitochondrial genome. So looking at this haplotype network here, blue represents the Plasmodium nozi genotypes in humans, while green represents the Plasmodium nozi genotypes in the macaques. You can see that there are no specific clustering of Plasmodium nozi between human and macaque isolates. In fact, if you notice there are also genotypes that are shared between the shares by Plasmodium nozi isolated from macaques and also from humans. This suggests that confirm that the infections of Plasmodium nozi in humans are actually a zoonosis, which is from macaques to the humans, right? So here we understand that zoonotic malaria transmissions, where actually the nozi parasites is maintained in the wild among the macaque populations, and how human got infections, where is by the mosquito that have fed on the macaques and bite the human as well. So basically, this is what have been confirmed based on the mitochondrial genome. Right. So the study has been expanded not only by our team, but also others from the Southeast Asian teams to studies about the Plasmodium nozi in the non-human primates. Now it is understood that the long-tailed and pigtailed macaques are the natural host of this Plasmodium nozi. Studies found in southern Thailand, the stump-tailed macaques and also dusky leaf macaques are also a potential reservoir for this Plasmodium nozi. This has been confirmed by using molecular methods like PCR and also DNA sequencing. So this suggests that Plasmodium nozi is not only confined in the long-tailed and pigtailed macaques, but also in other non-human primates as well. And it is largely distributed across Southeast Asia. Okay. Just a quick informations that I would like to share with you, that not only Plasmodium nozi that can cause zoonosis in humans, there are also cases of other Plasmodium, semen Plasmodium parasites, for example, like Plasmodium cynomogae and also Plasmodium inuii and corneae. There are also has been confirmed of natural human infections by these Plasmodium species, and all of these were confirmed by using molecular methods and also PCR and also DNA sequencing of the specific genes. Right. So, however, compared to Plasmodium nozi, the infections of Plasmodium cynomogae or corneae and inuii in humans are not as massive like Plasmodium nozi. So this is some question that we keep on raising. Why? Because you can imagine that they are of the same macaques. They are in the wild, but we have more Plasmodium nozi infections compared to this species inspection. So there's a lot of questions here as to need to be answered from time to time, and of course the research is ongoing to understand why nozis are more cases of in humans compared to this Plasmodium species. Right. So it is very interesting that these Plasmodium nozi infections are confined in Southeast Asia, and you don't actually see them in other countries like in China or India or any part of the world. Although there's a few papers, let's say as in the Europe, where people actually got plasmodium infections, but these people actually travel to Southeast Asia and they got the infections and they come back to their countries and then they're diagnosed with Plasmodium nozi. So Plasmodium nozi infections is actually confined in Southeast Asia. If you go back to the history of the evolutionary history of these parasites, where it actually coincides with the Sunda shelf, which was approximately 18,000 years ago. So nosai is an ancient parasites where it has been estimated that the most recent common ancestors was dated uh, roughly around 98,000 years ago up to 478,000 years ago. So these particular parasites has been involved uh, in Southeast Asia, how it relates to the movement of macaques and the movement of humans before the last ice age. And then when the Southeast Asians were and when the sea level rose up and developed, and of course the emerging of all of the islands that we see today, so you would see that there would be a limited movement of these macaques, of these all and these humans. And then from this onwards, we can think about the parasites, how they actually whether they evolve independently based on these geographical changes. Right? So because Plasmodium nosai is mainly confined in the macaques, when the sea level rise, so you would expect there is a less movements of the macaques, let's say between the mainland Asia to the islands of Indonesia and also in Borneo and the Philippines. So they the question would be, could they evolve independently and can cause exhibit different phenotypes when it comes to the infections, right? So from here, there is where we would understand the population genetics has come in, and to use this knowledge to understand the transmission dynamics of this nosai malaria. Right. So there's a lot of questions that can be asked due to this the history, the evolutionary history of this parasite, and also the related to this Sunda shelf as well. So one of the big questions here that we can ask ourselves, is there a genetic differentiations of Plasmodium nosai within Peninsular Malaysia or the mainland Asia, and also with the Malaysian Borneo as the natural host, geographically separated by the South China Sea. So we can hypothesize that due to the differences there would be a genetic differentiations, because how once they're separated, could they evolve independently, right? Another question is that if penozyte is an ongoing zoonosis, should there be no major barrier of gene flow in Plasmodium nosai from macaques to humans? Meaning to say that infections from in the macaques and also infection in the human, there should be no difference in terms of their genetic profile. So this is about the questions number 2. So if there is no difference, so it is an ongoing zoonosis, so there will be no human-to-human transmission, so there will be no adaptations in humans where it can actually just require human host without monkey host. So this is about questions number 2. So to answer these questions, so we first started analyzing to study this population genetics using slightly cheaper methods, which is using these microsatellite markers. Right? For these microsatellite markers, for those of you who are not familiar about microsatellites, it is actually a short tandem repeats that are found abundantly in the genomes, in eukaryotes, in humans, in Plasmodium. So the most common microsatellite genetic markers that we use are usually dinucleotide or trinucleotide, for example, like TAT or GAC, and we look at the numbers of the repeats and how these numbers of repeats, are there any differences within the population. So this is about the use of microsatellite markers. So when we design these microsatellite markers, we base on a different loci in the genomes where they are or evolve naturally in the genome, where they are not under pressures of, let's say, or drug pressures and so on. Right? So the location of these microsatellite markers that we choose, there are 10 microsatellite markers located across the Plasmodium nosai genome. So just an information that the gene that we choose these 10 microsatellite markers so that it can represent the whole chromosomes of the Plasmodium nosai. So once we design these microsatellite genetic markers, so we tested it on the human nosai infections in capid division. So we started from capid divisions here first, and using this analysis, we found out there's in capid, the nosai infections can be divided into two subpopulations. So we call it a cluster 1 and cluster 2 in infections. So when we apply these microsatellite genetic markers in Plasmodium nosai isolated from the macaques, right? We see that cluster 1 are mostly associated, are found in the Macaca fascicularis or the long-tail macaque, and cluster 2 infections are exclusive in long pigtail macaque or Macaca nemostrina. Now, this is very interesting because when we did that, we didn't actually expect that there's a genetic differentiations of Plasmodium nosai within the macaque populations, because you can imagine in the forest, these macaques are in the forest, they are exposed to this forest mosquito. There will be then intense transmissions. So because of the intense transmissions, you would expect there is no differentiations of the of nosai between and within this within the host. But when you look at this, there is a great differentiations of Plasmodium nosai between these two hosts, and these also seen in the human populations as well, in the human infections. Now, whether or not these population structures applies in other parts of Malaysia, so we apply this microsatellite analysis to Plasmodium nosai infections across Malaysia, different regions in Sarawak and different regions in Sabah states in Malaysian Borneo, and also some from the Peninsular Malaysia. And here we got more interesting results. If you look at the Malaysian Borneo, you can see the existence of cluster 1 and cluster 2 infections are maintained, are clearly seen within the Malaysian Borneo. You can see, for example, like capid divisions in the Betong, in Kanawit, in Sarike, and so on. So you can see that majority of the infections in Malaysian Borneo are related with cluster 1 infection, which is the Macaca fascicularis host. And then the infections of nosai from the Macaca nemostrina host are less common. But if you compare with the Peninsular Malaysia, it has a different unique Plasmodium nosai structure there, which is completely isolated, separated from the Malaysian Borneo type. So this is actually as expected. As the reason why I say expected, because how the nosais has been separated from Peninsular Malaysia, from the mainland Asia, and also from Borneo, so they would evolve independently. Right. So to confirm this population structure, they were analyzed using this microsatellite analysis, right? So we did an advanced analysis using the whole genome sequencing of Plasmodium nosai from patients across Malaysia. So we apply this whole genome sequencing and did this dendrogram of this using this whole genome data. So where we manage to get approximately 1.7 million SNPs to analyze. And using this neighbor joining dendrogram, So the existence of these three different subpopulations confirms supporting the evidence of this divergence as what we have seen by using the microsatellite analysis. Right? So in which the cluster 1 and cluster 2 subpopulation of nosai infections are strictly found in the Malaysian Borneo, while the cluster 3 infections are exclusively found in the Peninsular Malaysia, or at least I would say in the representing the mainland Asia. Right? And cluster 1 is infection is associated with the long-tailed macaques, Macaca fascicularis, while the cluster 2 is associated with Macaca nemostrina, or the pigtail macaques. Now, looking at how divergence, level of divergence among these three subpopulations, you can see that there are higher divergence since where we see using this fixed section index. A fixed section index, or FSTs, is calculated from 0 to 1, where 0 means there is no difference between the two subpopulations, while 1 means they are completely fixed. So this fixation index actually is a good index to measure population differentiations due to the genetic structure, as what we have seen here. There's a very simple formula that you can actually calculate this fixation index. Now, looking at this, when we calculate this fixation index among these three different subpopulations, you can see there's a higher divergence of FST when you compare population of Peninsular Malaysia and also populations of the Malaysian Borneo. For example, like cluster 1 versus cluster 3 and cluster 2 versus cluster 3, these allopatric populations. So this is as expected because the cluster 3 are separated from the Malaysian Borneo subpopulations by the Southeast Asia, by the South China Sea, by the South China Sea. However, we also see a high divergence between the cluster 1, cluster 2, here the FST is 0.32 in this table. Now, this is also very interesting because we didn't expect that. Initially, there's a level of divergence between this cluster 1 and cluster 2, but when we calculate this confirms there is a level of divergence within these in these sympatric populations, right? Now, due to this, we go into depth to look at the genetic diversity of these three different subpopulations. Now, we want to understand why we're doing this, because we want to understand, Plasmodium nosai is just initially when we started, it's just a Plasmodium nosai that's monkey to human. But now there's a different level of divergence of Plasmodium nosai in this human infection. So we want to understand why are they so different of Plasmodium nosai. Why do we see these three different types of Plasmodium nosai? So we look at the genetic diversity within these populations. Now, we examine the nucleotide diversity from chromosome 1 until 14. From chromosome 14, there are 14 chromosomes for Plasmodium nosai, right, from Plasmodium. You can see that there is a similar level of diversity if you look at the plot colored blue, cluster 1, and plot colored red for cluster 3. A similar level of diversity in cluster 1 and cluster 3 subpopulation. You can see that the pattern are similar of cluster 1 and cluster 3. Now, this tells us a little bit funny because cluster 1 and cluster 3 are totally separated by the South China Sea. But in fact, when you look at the level of diversity across these chromosomes, across these genomes, they show a similar pattern, a similar level of diversity. Now, when you look at the cluster 2, or cluster 2 infections, which is associated with the with pigtail macaque, you can see there is a reduced level of genetic diversity in these cluster 2 populations. Now, what does this mean? It means that cluster 2 infections, the formation of the cluster 2 in parasites are a result of a bottleneck from within the pigtail macaques. Right? So these initial bottlenecks, where it start to develop in these pigtail macaques, right? So you can see the level of the genetic diversity will reduce. Right? So it will maintain. It is fully adapted in the pigtail macaques. That's why you can see there is a reduce of genetic diversity. Right. So this is a very interesting about the level of how different cluster 1, cluster 2, and cluster 3. Right? Although they are separated by the host and also separated by the South China Sea. Now we go to the next step, which is to look at the divergence, the genomic divergence across the different chromosomes. Right? And also this is used by measuring the fixed sections index, like what I have mentioned just now, which is from 0 to 1 across the chromosomes. So we compared, we do pairwise comparisons here. So we compared cluster 1 infection versus cluster 3 infections, cluster 2 infection versus cluster 3 infections, and cluster 1 infection versus cluster 2 infections. When we look at the comparisons between the divergence between cluster 1 and cluster 3, we can see there is a homogeneity of this level of genomic divergence across the genome. You can see that the patterns are not that the difference are not the differences are not that obvious. So we say this there is a homogeneity. But when you look at cluster 1 and cluster 2, which is within the Malaysian Borneo itself, you can see there is a highly divergence, a level of divergence within the Malaysian Borneo itself. Now, this is very interesting. Although they are of a symmetric populations, okay, you can see there is a highly divergence of Plasmodium monosi between found in from the pigtail macaques and the long-tailed macaques, right? So this is a little bit unexpected across the genome, because you would expect that there's an intense transmissions in the by the mosquito, but there is something like a restricted gene flow here between the cluster 1 and the cluster 2, between the pigtail macaques and the long-tailed macaques. So we go into the detail across these 14 chromosomes, which of these chromosomes has a particular sites of higher divergent region. So here we illustrate this, what we call by genomic islands of divergence of this cluster 1 and cluster 2 infections from Malaysian Borneo. And we can see that there is a huge and a very obvious divergent regions found in chromosomes 7, 12, and 13. Right, so the chromosome 7, 12, and 13 actually is a very you can see this how different are cluster 1 and cluster 2 due to the higher divergent regions found in these 3 different chromosomes. You can also see that based on these calculations, we found that 28% of the whole genomes are located in the highly divergent regions. So that's why cluster 2 and cluster 1 and cluster 2 in Malaysian Borneo, they are different due to the existence of these genomic islands that are across the genome here, colored in red here. Okay, so this tells us that in our early understanding of this zoonotic infection by this Plasmodium monosi, we understand this is the original illustrations of how the transmission occurs. But because of the analysis that what have been described just now, it is understood that there is a two independent Plasmodium nozae zoonosis are occurring in Malaysian Borneo. At least two independent zoonosis here. Why I would say at least? Because we see here, we only knows that these are based on two different macaques, pigtail macaques and long-tail macaques. We haven't explored other possible monkeys in Borneo yet. So here, at least we understand that cluster 2 and cluster 1 and cluster 2 infections are two representing two independent zoonoses that are occurring in the Malaysian Borneo. Associated with two different macaque species. Now, the big question here is that is there's a transmissions of this plasmodium uh, occurs between long-tail and the pigtail macaques by a specific mosquito vector here. So these are the questions that are still ongoing in our analysis to find out whether the vector is the one that's actually restricted the flows of nozae between these two macaque species. So, right. So now we understand the transmissions dynamics. How complex is the transmissions of Plasmodium nozae in the Malaysian Borneo? So we understand that at least there are two independent zoonoses. What about the rest of the Malaysian Borneo? Right. So based on our early microsatellite analysis, we know that across the Malaysian Borneo, there are also existence of the cluster 1 and cluster 2 infections. So our next step, due to this two independent zoonoses, we would like to see, using this and genomic data, we would like to see whether cluster 1 and cluster 2 are occurring across throughout the whole Malaysian Borneo. So doing whole genome sequencing is very expensive. So that's why we develop a very simple PCR tools, genotyping tools to identify this cluster 1 and cluster 2. So we identify a fixed SNPs from the whole genome data, where we develop two simple single round PCR assays to identify cluster 1 and cluster 2 infections in humans. So these cluster 1 and cluster 2 genotyping tools are actually only applicable in the Malaysian Borneo, because we don't find these cluster 1 and cluster 2 infections in Peninsular Malaysia. So these genotyping tools is very cheap and can genotype all Plasmodium nozae infections in Malaysian Borneo. So using these genotyping tools to understand the prevalence of this cluster 1 and cluster 2 infections, so we tested it in our archive samples that we collected since 2000s from 3 different study periods. And based on this, it shows that cluster 1 infection has been a dominant types of nozae infections in humans in capit divisions, followed by cluster 2. We also see a small portions of humans were infected by both cluster 1 and cluster 2 parasites of P. nozae, but the number is very, very low here. Right. So we also look at the proportions of these Plasmodium nozae infections across the 5-year study period to look at the pattern of this cluster 1 and cluster 2. So the period of studies from 2013 to 2018, where we have complete P. nozae cases obtained from this Capit hospital, right? So across these studies from 2013 to 2018, it is confirmed that the cluster 1 infections are occurring 1/3 of the total infections. So it is maintained that cluster 1 is the predominant types of Plasmodium nozae that is infecting the human compared to cluster 2. Right? And then we also see, using this temporal and trend patterns analysis, we also see the patterns of the cluster 1s, how cluster 1 infection actually distribute to the total numbers of Plasmodium nozae infections in humans. So this shows that confirm the cluster 1 infections are predominantly found in the human infections, and these humans are naturally exposed to the natural host, which is the Macaca fascicularis, for acquiring the clinozine infections. Okay, so we also tested these genome mapping tools in all human infections using our respective samples from 2001 until 2013. So we found that cluster 1 infection are very dominant across Malaysia and Borneo. But we also can see that cluster 2 infections are high in certain regions in Sarawak itself, which is in Miri. Miri is very near to Brunei, and also in Kapit divisions in Sarawak. In Kenawit division in Sarawak. Right? So these different prevalence of cluster 1 and cluster 2 infection across Malaysia and Borneo can be contributed by many factors actually. It can be contributed by the environmental changes, for example, like deforestations, and then these also can be distributed uh, due to the changes of the macaques populations distribution and also the vector behavior. So because of this, we can see that's why some regions have higher level of cluster 2 infections and some have higher cases of cluster 1 infections. Right. So when we present this data, of course, clinicians in the hospital, they are more interested in the treatment, right? So we look at we compare the clinical, laboratory, the epidemiological features of these two divergent subpopulation of Plasmodium monocyte. So based on our studies, looking at the patient's presentations or the clinical presentations, the laboratory data, looking at the assessments of their blood tests and so on, hematological tests, lipid profile, and so on. And we see that there is both cluster 1 and cluster 2 infections have similar disease phenotypes, meaning to say that they present the same level of clinical presentations, severity, and so on. Therefore, there is no changes in the clinical management when it comes to this cluster 1 and cluster 2 infections. But when we look at in terms of public health point of view, when we look at the distributions, the hotspots of this cluster 1 and cluster 2, so we did this analysis in the Kapit division itself, based on the infections patients, we have forest activities 2 weeks prior to their hospital admissions. We can see there's a changes of hotspots of cluster 1 and cluster 2 infections across this year from 2014 here until 2019 here. Right? So you can see that most of the infections occurs. You can see the difference between the cluster 1, cluster 2. You can see that most of the infections of the cluster 1s are widely distributed, and the hotspot is larger than the cluster 2 infection. That's why you can see that most of the cases are associated with the cluster 1 infections. So when we look at the risk activity compared from between cluster 1 and cluster 2, we can see that cluster 1 are mostly associated with small-scale farming and the longhouse activity or the village activities. Meaning to say that this small-scale farming is where they do some small farming near their longhouses, near their village, right? So this is where they are exposed to these monkeys nearby, because macaques are preferred, especially long-tail macaques, are preferred to be near the human settlement, especially at the farm where they can have easy access to food. So that's why most of the cluster 1 infections patients are related to this small-scale farming and the longhouse activity. And of course, if you look at the sex, so maybe between male and female, for cluster 1, they are more or less 50%, 50%, or they are not much different. But cluster 2 infections, you can see that most of these people who had these infections by cluster 2 types of P. nosai are associated with deep forest activities such as hunting, logging, laborers, or deep plantations. And most of these patients who are infected by these cluster 2 nosais are male, because most of males are doing this kind of activities like hunting and logging compared to female. So these are the two different risk activities, and also the characteristics of the patient when it comes to cluster 1 and cluster 2. So this particular actually can give us an idea about how we can actually do our interventions in the community about how they are exposed to different types of pinocyte infections. Right? And furthermore, for cluster 2, because it is associated with the pigtail macaques or Macaca nemostrina, we have to understand that the characteristics, the behavior of Macaca nemostrina, this particular monkey is very reserved. They prefer to be in the deep forests, away from humans compared to Macaca fascicularis or associated with this cluster 1. So that's why we understand why these risk activities, when we relate it with the macaque host and the human infections. Okay. So this study, where we publish it in this scientific report, a journal, that we conclude the cluster 1 infections of nosai are related to the small farming activities near the residential areas, while the cluster 2 infection, which is associated with the Macaca nemostrina, usually confined with the deep forest activities such as logging and hunting. Okay. Now, this is almost the end of of my talk. So, of course, when we want to understand the transmission, we cannot neglect the mosquito vector as well. So, previously, we have incriminated that the Anopheles of the Leucosphaerus group in Sarawak are the main vector for this Plasmodium nosai, where these vectors breeds in ponds, in rain pools, and they are forest mosquito. They are also found in forest fringe and farm. And these particular mosquitoes feed outdoors on both humans and macaques, and they prefer to feed from 6 p.m. until 6 a.m. At night. So this is the understanding that we have been that we know for the past 15 years. But recently, my PhD student have found that the vectors are actually not only confined in the Anopheles of the Leucosphaerus group, but also expand to other types of groups. For example, like the Babesiosis group, the Tessellatus group, Mucolatus, and Umbrosus. So you can see that in the carpet division itself, that there are different species of Anopheles can carry a different Plasmodium species from the macaques to humans and increase the chances of zoonosis in humans, especially when the humans perform activities in the forest or in the farm. So there are higher chances of getting infections because there are a lot of Anopheles species that can actually transmit these parasites from macaques to human. Right. So the identifying of these Plasmodium species were used by both molecular method using PCR and also confirmed by DNA sequencing of target gene. Okay. So I think this is the last one. So I'm sharing with you not only that we studied the nosai infection, zoonotic malaria in Sarawak, we also try to understand the malaria infections in the Kalimantan area that are bordering this Malaysian Borneo. So my PhD student who's graduating this soon, Dr. Diana Natalia from Untan, right? So she has collected these samples, human samples across in three provinces: West Kalimantan, East Kalimantan, and North Kalimantan, and in these different regencies. So we found that the Plasmodium nosai are not only confined in the Sarawak, but she also found that the infections also found in communities particularly in the West Kalimantan. Okay, so samples were collected from both febrile and non-febrile participants across these study sites, and using PCR and DNA sequencing, so there is an evidence of Plasmodium nosai infection and Plasmodium innoi infections, particularly in the West Kalimantan, in Sanggau, Sintang, and also Kapuas Hulu regencies. Right? So this is a very interesting studies, because the reason why we did this, because all this while we have higher cases of malaria or zoonotic malaria in Sabah and Sarawak in Malaysia and Borneo, but there is a very small cases, very rare cases of this zoonotic malaria in Kalimantan. So that's why we did this, to understand why if there's actually evidence of nosai and inui that are areas sharing their borders of between Sarawak and Kalimantan. Okay. So, as a summary of these studies, we know that there are at least three highly divergent of Plasmodium infections occurring in Southeast Asia, with cluster 1 is associated with the long-tailed macaque host and found in Malaysian Borneo. Cluster 2 is associated with the pig-tailed macaque host found in Malaysian Borneo also, and cluster 3 are exclusively found in Peninsular Malaysia or mainland Asia. And then we found that there's an occurrence of at least 3 independent zoonoses overall of Plasmodium nosai. And then the infections that is related to long-tailed macaques are very common, and different outdoors and forest activities are associated with different types of nosai infections, particularly in the Malaysian Borneo. Before I end, because these nosai infections are solely in zoonosis, could there be a challenge for zoonotic malaria eliminations? Right. So just to brief, to tell you that the Malaysia have been no cases of Plasmodium human Plasmodium species infections since 2018, but we have continuously high cases of zoonotic malaria since 2018. So because of this, our malaria-free certification has been put on hold until we can reduce the zoonotic malaria infections in Malaysia, right? So since it's a zoonosis, there's a lot of things that can need to be answered here. For example, like changes in the ecology for during logging and agriculture, the vector species and the behavior of the vector and the human populations. Due to this active deforestations and also agriculture, that potentially increase the infections. So could urbanizations is the best answer to eliminate zoonotic malaria? Okay. Different types of Plasmodium nosai, could it lead to adaptations in humans in the future? As what we have seen that these species are adapting very well in the pigtail macaques. All right. So with this, I would like to end with a lot of peoples that evolved in these studies throughout this more than 20 years, actually, when we first started. So these are the teams that are actually working together. This is the local team, and also some of the visitors from Indonesia and also from the UK joining us, and also other universities, local and international agencies that is work together with us. So with this, I would like to end my talk, and I am happy to answer any questions from you. Thank you very much. Yes. Oh, we have one Ria Star, Irina.
Okay. Thank you very much, Professor Paul, for the insightful and comprehensive presentation that you have clearly highlighted how zoonotic malaria is not just a biomedical issue, but also a socio-ecological challenge. And Professor explained the complexity of the transmission, such as how human activities and then environmental changes and animal reservoirs are all interconnected. And this perspective is important because it reminds us that controlling zoonotic malaria requires more than medical treatments, including policy making. And Professor given us a broader understanding of why tackling zoonotic malaria is both urgent and complex. Okay, so we begin to the discussion session. I will open the floor for everyone who wanna ask the question, and if it's not possible, you can. If it's not possible to open your microphone, you can type the question in the chat box. So there will be question per term. Let's start the session. So you can raise your hand and also introduce yourself. All right, so let's take a look. All right, for Miss Ariesta. Hello. Could you turn on your microphone? Don't forget to introduce yourself and ask the question.
Wina
Kok
mana
nggak ada?
Wina?
Kok nggak ada? Ariesta?
Okay, so before the host haven't give me the access to open my microphone. Okay, is my voice already clear enough?
Yes.
Okay. Thank you so much for Professor Paul for giving the outstanding presentation about the transmission of zoonotic malaria. I have my question here, but before I want to share first that currently in Indonesia there's upcoming agenda to live up against the X transmigration area, which include 154 locations all around the country, and it's including the endemic areas like Papua and Kalimantan. But the problem is, and what's make it interesting is that I found that many of those locations still have inadequate prevention and surveillance regarding malaria, and it's due to many difficulties like access, tools, and support, and also the funds, and also the human resource too. But what do you think what the community itself can do to strengthen the prevention of malaria? And maybe Professor Paul, do you have any more study cases that maybe we can learn from to this matter, especially about the community-based practice in malaria prevention in Lakso results for area? Maybe that's my question. Thank you.
Okay, thank you very much for your questions. Now, community-based. Now, when it comes to zoonotic malaria, at least for what we are applying in Malaysia, um, all this while, the ministry have been distributed, like bed nets and so on, and giving talks and doing surveillance, like blood tests, microscopy, and so on. But the cases of zoonotic malaria has still, steadily high. So currently, the best way to actually to reduce the number of zoonotic malaria is actually by education, health educations, where we need to actually inform the community to protect themselves, especially when they are going engaging in activities like forest activities and so on. But however, zoonotic malaria is something that is hard to actually to control, because how they expose themselves in the forest or in the farm, especially on getting bitten by the mosquitoes. So one of the important things here is that the humans' behavior that need to be changed. Sometimes people are neglecting about, safety, protect yourself from getting bitten the mosquito. Of course, you can see people can cover themselves, but when it comes to hunting, people doing like hunting activity, so one of the thing that when we ask them about how do they protect themselves, they said that they cannot use insect repellent because the smell of the insect repellents can actually attract, can be sensed by the animals, and so they cannot do hunting. So, from this, it is very important that the community itself need to actually understand how the infections occurs and how they can protect themselves. Besides, if they do not want to use insect repellents, so they should protect themselves and covering up. Now, this is about human behavior. This is very social science need to work for there. And other thing is that when it comes to the malaria in general, of course, malaria in general, human infection by the human Plasmodium species, you can see that it is possible to actually, to cut down the infections by massive bed nets, in the apa, using the insect repellents, protect themselves, and so on. Another thing that is very important when it comes to zoonotic malaria here is that not only that we want to reduce the infections, we also want to reduce the death, because the death cases by Plasmodium nosai. Plasmodium nosai, it actually is a very fast parasite when it replicates in human. It takes 24 hours to actually to multiply in human compared to other Plasmodium species. So delayed treatment of Plasmodium nosai, okay, will lead to death. We got lead to severe malaria, multiple organ failures, and will lead to death. So this is the main in Malaysia, for at least in Malaysia here, we try to to have zero death cases by malaria. This most important thing, followed by the reduce the cases of zoonotic malaria, because zoonotic is very difficult to reduce because of the exposure to wild macaques, to the mosquito in the forest. But at least in the community, the best is actually for at least in Malaysia, is by health educations, reminding them to protect themselves. Report when you have fever, and so on. Get treated when you have fever, and so on. Yeah, and the government, the surveillance, is also plays an important roles here. Because from the surveillance, human surveillance, where we can see the malaria burden in the area. So in Malaysia, we use microscopy as our gold standard. We don't use RDT and so on. We use microscopy, but it requires a multiple collaboration with university as well to actually use molecular methods to help to confirm the infections in the community as well. Yeah. So I hope I answered your questions.
Okay, thank you so much, Professor, for your insightful answer. Thank you for having your time with us.
Okay, thank you so much for Professor Paul for the answer and the question for Miss Ariesta. Okay, next for the second question to Miss Raisha. Could you please turn on your microphone? Yes, please.
Okay, thank you, Nabila. Thank you for the opportunity. Okay, Prof. Paul, I wanna ask about your slide. I think it's third or fourth slide. Is it about the Plasmodium infection in human, which is considering that Plasmodium knowlesi is often misidentified as Plasmodium malariae under microscopy. Is it also possible that PCR assays using RMIL1 and RMIL2 primers for malaria may actually amplify nuclei due to sequence similarities because which is the RMAL primers is the specific primer for Plasmodium malaria. Okay, thank you.
Okay, all right. Thank you very much for your questions. Yeah, the primers for Plasmodium malaria and also PCR primers. You're talking about PCR primers, yeah? And the PCR primers for Plasmodium nosai that what we have, they are now very specific. There's no cross reactions. Now, previously, when they report Plasmodium malariae from this human infection was because of based on microscopy. Maybe I missed that. It was firstly reported by microscopy, or so looking at the morphological characteristic of the parasite under the microscope. So that's where during that time they reported as Plasmodium malariae, because during that time there was no molecular method to identify malaria and nosai by using PCR. Now, due to that, so that's why the team developed the molecular PCR methods to identify Plasmodium nosai, and then and also PCR primers for Plasmodium malaria to distinguish these two different species, right? So I should have put just an introduction that the morphology of malaria and nosai are very similar. Right? Under the microscope, you cannot tell the difference. Even the experienced microscopists cannot tell the difference between the two species, right? Unless you put another element or information like travel history and risk and activities. In CAPIT, when the patients come and when the patient come with malaria, and the doctor will ask their travel history first and also their activities, what they have been doing for the past 2 weeks, right? So with that, they can see whether the patient will be, will be having infected by Plasmodium nosai or other Plasmodium species. So when the report come from the laboratory based on the microscopy, right? This is what we're doing because microscopy is still a gold standard in the Ministry of Health facilities here. So when the report comes, they look a band form under the microscope which represent malaria or nosai, right? So the doctors we combine the activities or the travel history, whether the patient is plasmodium affected by nosai or malaria. Now, this is the thing I just want to share with you. Sometimes in the hospital, the microscopists may always mix infection, right? So this is where sometimes where sometimes they send sample to the university for us perform PCR to identify to confirm the species. So sometimes we found that the patient who have P. nosai, so sometimes they mix with Plasmodium vivax, or patient come with P. vivax. So when we do PCR, the patient have an infection of P. vivax mixed P. nosai. It means that the patient have a travel history from vivax endemic countries. Okay, so when they come back to Sarawak, they go to the forest do hunting or farming. They're exposed to the monkeys. They also got infected by Plasmodium nosai. So sometimes this is what has been a bit alarming, because you tend to mix infections when it comes to microscopy. So molecular method is still actually the best to confirm a species, but it is expensive. PCR machine is very expensive, right? And the reagents. Yeah. So I believe in Indonesia, RDT is widely used there. So there's a pro and cons RDTs and microscopies, and also PCR, right? But in Malaysia, so we use microscopy. We don't use RDT here. But to confirm it, they will perform PCR. Right. Okay.
Okay, thank you, Professor Paul.
Okay. Thank you very much, Professor Paul, for the answer, and to Miss Raisha for the questions. So we are before we move on to the next session, to the uh, next question, allow me to read the question in the checkbox. Okay. Okay, Professor. Thank you, Dr. Paul, for the insightful presentation. So this question is from Angela Priscilla Helen, Miss Angela. I mean, so sorry. Okay. She have question is genetic diversity among Plasmodium knowlesi can affected to clinical manifestation on malaria disease. And for other Plasmodium macaca, is it possible in the future have ability to transmit it constantly in wide area like Plasmodium knowlesi? Thank you, Dr. Paul.
Okay,
So
so
that's the question.
right. So there are two questions here. So the genetic diversity among pinocyte can affect the clinical presentations. So like what we have published, I'm showed you just now. So we look at the when we look at the two different cluster 1 and cluster 2 of Plasmodium nosai, so we look at the clinical presentations, the laboratory, the lipid profile, liver function test, as the severity. So there is no difference in terms of the clinical presentation of between these two genetic diversity of Plasmodium nosai, at least what we see in cluster 1 and cluster 2 in Malaysian Borneo. Therefore, there is no difference in terms of clinical management. Right? So the answer for this is that there is no effect on the clinical presentations. They both present the same clinical presentations. For the second part of the questions, or about other plasmodium from the macaca monkeys, the macaque monkeys, is it possible in the future have the ability to transmit constantly? Well, in theory, what we see in nosai, in theory, yes, they should be able to be ability to be transmitted constantly like P nosai. The big question here is that why their number of cases like Cynomolgus inui, the cases are very low compared to nosai. So in theory, yes, you can see that there's a possibility, but here we need to explore further as to why are these other plasmodium species the there is a very less transmission from the macaques to humans. Only one or two in, let's say, in 1,000 cases, maybe two, three are positive for cynomolgi or in wine. So here it involves many different factors. For examples like the immunological status of in within this mosquito vector, right? And also could there be some species of Plasmodium are more dominant in the mosquito, and then it will suppress other species or other genotypes? Or these are the questions that we are actually trying to answer, and the research here is still ongoing as to understand why there is less cases of other Plasmodium species compared to Plasmodium nosai in human. Yep.
All right, thank you. Thank you, Professor. Is there any feedbacks or following question to Miss Angela? If there is not any feedbacks or any following up questions, so we're moving to the next question. And for everyone, I will make a discussion session alternately in the chat box and those who have raised your hand. So we will move on to the who raised hands. To Mr. Elias, could you please turn on your microphone? Yes, please.
Okay. Good morning, Professor. Thank you for the insightful presentation. From the many challenges we had based on molecular perspective, what research holds the most promise for understanding the future genetics evolution of Plasmodium knowlesi? Okay, thank you.
Future studies of this. Okay, now there's a lot of potential studies when it comes when it comes to the genetics or genomic studies to actually to understand of about these transmissions of this zoonotic malaria. You can see it at different angles, like for examples, like about especially from the parasites in the mosquito, for example, right? So different genotypes in the mosquito, how they actually survive in the mosquito and not being transmitted to humans. Right? So these are examples by looking at the different types of parasites, looking at the diversities of the parasites in the mosquito once they have been fed in the macaques before they can be able to be transmitted to humans. So those are another example because we want to understand why certain genotypes or certain species are easily transferred from macaques to humans and why others are not able to be transmitted. So the other possibilities to answer is all come back to the research, to the questions that you want to answer, want to understand about the transmissions, right? When we describe this cluster 1 and cluster 2 and this cluster 3 from this mainland Asia, we do not understand whether these two types of parasite, cluster 1 and cluster 2, from these two different macaques, can they actually mate? You know, the gametocytes, can they actually breed between the two, or they just separate? Right? If they cannot like mate, of these two parasites of non-zyke of cluster 1 and cluster 2 parasites cannot mate, could this represent two different species actually? Right. So this need further cell biology analysis to understand about this cluster 1 and cluster 2. Another thing that research can be done about is that besides the two macaque species, are any other macaques or monkeys in the wild, right, carry these parasites, and are there any other simian Plasmodium species are found in the forest? Because you see, forest is a house of many animals, many primates, right? We cannot explore all of it because they are in the wild, and it's difficult to catch them. At least for orangutan, they are exposed. You know, uh, are the Plasmodium species from the orangutan, can they be transmitted to human? Because orangutan and humans are of the same, you know, in the animal kingdom, are evolutionary closely related lah. You can say like that. Could their parasites also be able to adapt to human as well? Yeah, this is an examples. Right? So there's actually a lot of questions when it comes to this. All of this require genetic and genomic analysis to understand the parasites transmissions. Right? And of course, others would be, besides genetic, you can go beyond serology, right? People are exposed, how people are exposed, has they been exposed, even though they don't get sick, are these farmers or people work in the forest, are they actually exposed to malaria? And if they are exposed to malaria, they got malaria at some point, but they don't present any clinical signs and symptoms, all right? Could they actually develop immunity towards them, natural immunity? So there's a lot of question actually can be, you know, can be explored from this zoonosis, the term zoonosis here. Okay.
Okay, thank you very much, Professor Paul. So, is there any feedback or following up question from to Mr. Elias? Okay, thank you so much. So we're moving to the next question in the checkbox. Allow me to read it. Good afternoon. Let me introduce myself. My name is Karisma, a master's student in epidemiology.
I have previously worked on malaria control in Papua, but I have never had direct contact with malaria caused by Plasmodium knowlesi. Could you please explain the most characteristic genomic differences between Plasmodium knowlesi and other Plasmodium species? Thank you so much. That's the question.
All right. Thank you very much, Karis Mata. Okay, now this is very interesting. In Papua, if you understand, I think there's a paper that by Professor Babir Singh and Professor Janet Cox saying that, this Plasmodium nosai, it has its limit when it comes to Indonesia. If you know, if you understand the Wallace line, okay, the Wallace line actually is an evolutionary invisible line where we restrict the macaques species and also the Anopheles. Some regions, for example, like Papua, there is no specific macaques, there's no specific Anopheles of the Leucosphaerus group for particularly for nose I found in Papua, not even in Sulawesi. Because of this Wallace line, that these animals that they are not found in this region. So that's why you don't see Plasmodium nosai there. This is the same thing occurs in India. We don't have Plasmodium nosai in India because there is no long-tail macaques or pigtail macaques there. And the macaques there, they have rhesus macaques in India are very aggressive, okay? So they cannot live together with the long-tail and pigtail macaques, so that we kill these other macaques. So these restrict the occurrence of Plasmodium nosai in India as well. So that's why in Papua you don't see Plasmodium nosai because there's no macaques, suitable macaques there, or even the vector due to this, which is answered by this Wallace line. If you Google Wallace line, you can see how the animals are actually restricted into certain areas, are not found in these regions. Okay. And these characteristics, this not because of the genomic differences of this Plasmodium nosai, that this is just because of the host, the specific host that they are suitable for these macaques. So that's why we also, when we work with our collaborators in Sulawesi recently, they don't find any nosai there, although they found other species like Inwai, but they don't find nosai because nosai are not found in particular different kinds of monkeys that area, and also the Anopheles there as well. The vector are not suitable, right? So in Papua, you still find other like human Plasmodium falciparum and so on, but perhaps you can probably explore using molecular methods like sequencing to see if there's any other potentials non-human Plasmodium species there, since I do believe that even the Papua itself have a different ecology, different animals, different primates that potentially, I don't know, this is just a theory for me, potentially can cause other types of zoonosis there. Yeah.
Okay, thank you so much, Professor, for the answer. So we're moving to the next question to Mr. Surya. Could you please turn on your microphone? Yes, please.
Okay, thank you, Nabila. Okay, so good morning, Professor Paul. So thank you for this opportunity. My name is Surya Negara. I'm from Health Entomology student. Okay, so my question is, like, for example, in a certain area of where malaria is endemic, people are infected with Plasmodium such as Plasmodium vivax or Plasmodium falciparum. Is it possible that this infection can trigger antibodies, but that also react to another species, like for example, Plasmodium knowlesi? Because I once read an article mentioning that in that area that only Plasmodium vivax and Plasmodium falciparum infection were reported, yet antibody testing indicated that the presence of antibodies against Plasmodium knowlesi were detected. How could that happen? And is it possible that Plasmodium knowlesi can be amplified by RMAL1 and RMAL2 primer? That's all my question. Is it clear?
RML. Okay. So, sorry, I didn't get your first your first part of the question. What was your first part of the question?
Okay, maybe okay, so like my first question is like, for example, maybe in like the region of area where malaria endemic is only caused by Plasmodium vivax and Plasmodium falciparum, is that infection can like develop another antibodies species like maybe for Plasmodium knowlesi antibodies.
Okay, so in certain area that is you can see like Vivex or falciparum, right? In this area, even you can see even in Sarawak, uh, in Kuching where I live here, you don't see much noza. You can see Vivex. Well, occasionally cases, very rare cases, but you can see Vivex or especially Vivex. It's just because that the vector or the macaque here, they don't actually carry Plasmodium nosai, or they're not infected yet by Plasmodium nosai, or there's not the parasite has not reached to this due to this area yet, right? So that's why you don't see any Plasmodium nosai infections. Whether or not they develop immunity to Plasmodium nosai, you need to expose first, natural exposure into a Plasmodium nosai to get an immunity towards P nosai. But because humans are not, a natural host for Plasmodium nosai, when humans are infected with nosai, they can easily get sick because they're not the natural host. Okay, but of course here we haven't explored. When we go to look at the indigenous community in, for example, in Sarawak, where in that area we can see some of P nosai cases, but we don't see. Sorry, we see P vivax cases, but we don't see P nosai cases. They're not very common, right? And this community, because they are like semi-nomadic, yeah, so they go to the forest, expose and serve, right? So they we believe that could be this particular indigenous community who are semi-nomadic have developed immunity towards Pinozai because they are exposed in the forest, right? But we haven't explored that in detail to confirm that yet, right? Because the question is very, very valid. There could they have developed immunity? Could P vivax also help? I actually cannot really answer that, but in theory, yes, they could, compared to other community, like say in Kapit, where they are doing farming and so on. So this community, they don't actually develop, but they get sick very easily, right? Although they are exposed to monkeys from time to time. Some patients, when we look at the database, some patients come two, 3 times to the hospital, okay, with the similar P. nosai infection. They keep on reinfected by P. nosai. So, but these are the certain community. These are the community. They are not semi-nomadic. They are not nomadic or semi-nomadic. These are the community that are macam they're already like stay in the kampong, in the village. But the semi-nomadic community that we actually haven't explored, it's quite hard to define to get their blood. Taboo and everything kan about this nomadic community kan. So we do believe that there could be uh, a level of immunity, natural immunity in them when they're exposed. Because it's funny when you think about, they're going to the forest from time to time, and the forests always have monkeys, but they don't get sick easily. But once in a while they do get Vivex. Where do they get Vivex from? We do not know. Yeah. So yeah. So another thing that can be explored is to study at this semi-nomadic or nomadic community, how they actually develop the immunity, and do they actually have this immunity towards Plasmodium nosai infections? Okay. Now, so your second questions regarding the primers. Okay, so primers are very specific. All this while, I would say the PCR primers are very specific. There's no cross reactions. Okay, so what we've been doing, like RML 1 and 2, they are very specific for malaria. They won't cross-react with P nosai. right? So we've been doing this for many years already. We keep on using it. There's no cross reactions. They're highly specific. Even for P. nosai primers, they don't actually cross react with any other species. So they are highly, how to say, reliable, I would say lah. Huh? All this while reliable, unless people have reported it. They are not specific. They cross react. Yeah, yeah.
Okay, thank you. I think that's all answered my question. Thank you, Professor Paul.
Okay, thank you so much. Okay, thank you so much for the answer to Professor Paul and also the question from Mr. Surya. So we next move on to the question. We have another question in the checkbox. So allow me to read it. This is from Miss Jia. Okay. Good afternoon, Professor Paul. Thank you very much for the insightful lecture. I would like to ask about Plasmodium cannabis. Why can Plasmodium cannabis infection in humans cause very high parasite levels in the blood and several symptoms, even though it is originally a zoonotic parasite from macaques? Okay, that's the question, Professor.
All right, thank you very much. So this is a very good question also. So as you see, nosai, this is the shortest, has the shortest erythrocytic cycles compared to other Plasmodium species. Erythrocytic cycles mean that it is in the blood stage. When they complete the life cycle at the blood stage, they have the shortest one to complete one cycle, which is 24 hours compared to other Plasmodium species. So imagine that 24 hours compared to Falciparum takes about 48 hours, Vivax 48 hours. You can see 24 hours, you can develop very high number of parasites in the blood. That's why it can cause high parasitemia if you don't treat the patients. Okay, now this high parasitemia also can lead to a severe malaria where patients can develop affect the organs like kidney and also lungs due to the immune response of the body to the parasites itself. So, but if you compare it, so that's why in humans with Plasmodium nosai tend to have high parasitemia. But this is funny when it comes to in the macaques, in the natural infections in the macaques. Now, we found that in the macaques, we see that high parasitemia is not common in the macaques, what we have observed throughout our studies. Sometimes we even have difficulties to find these parasites under the microscope from these macaques blood. So, when parasites live in the natural host, so they will not harm the natural host, so they will able to maintain the level of parasites in a very low level so that they will not harm their natural host. So that's why in the macaques, when people ask, sometimes people ask, will the macaque get sick when they infected with nosai? So when we ask our veterinary, they would say that the macaques are they don't present any sickness, illness, even though they are positive for malaria parasite. So this is how interesting that the parasites in the natural host can actually able to maintain their level as low as possible so that the macaque itself can actually do not develop any illnesses. But when it jumps to the human, because human are not the natural host, our immunity are not as strong towards P. nosai, so we get sick easily. So that when the parasites multiply very rapid within this 24 hours.
Okay, thank you very much, Professor Paul. I hope to Miss Jia get the answer from Professor Paul. Okay, because Dr. Fauzi raised his hand, so maybe you want to convey something or give a response. So could you please turn on your microphone? Yes, please.
Thank you, Nabila. Paul, I have a questions. Not a questions, but maybe some of the questions. It's my curiosity, Paul. So the first question is about the transmissions differences between Malaysia, Borneo, and Indonesia, Borneo. If we look at the population of the monkeys in Malaysia, Borneo, and Indonesian Borneo, they are actually sharing the habitats. They can come across between these two countries. But why? The question is always a to the experts like this. Why in Indonesia there are not many nolicide cases in Kalimantan? There are cases, but not that many as Malaysia. Is there any hypothesis that can cause the differences between the Malaysia, Borneo, and Indonesia?
Yeah. Okay, thank you very much. Yes, a huge difference if you look at the different aspect here.
Exactly.
So, my PhD student, Diana from Untan, so this is what her study is all about. Why there's a low cases of nosai in Indonesia, at least Indonesian border, right? Compared to Sarawak, compared to Kapitlah, for example, although a large portions of the borders are shared in these Kapit divisions with West Kalimantan at least, in Kapuas Hulu Regency. So now this go back to how we manage malaria cases, right?
Mm.
In Malaysia, every single suspected malaria will be recorded and then will be examined thoroughly by microscopy and so on. Okay, and then they're positive. They are they have to be admitted to the hospital until they are cleared for malaria. Okay, and every single case of these malaria patients in the hospital need to be notified. Otherwise, the doctor will get trouble from the ministry. Okay, every single case. So even we presume that the patients have fever, okay, so at least in Kapit, so they will go straight to the hospital and examine like every single cases now. In Indonesia, for at least in, let's say, in Putu Sibau at Kapuas Hulu, this was studied by Diana, right? In that area, people are still when people have fever, okay, so they will screen with RDTs, and then when they're negative, and that's it, they release, they are,
Yeah. Mm.
going back, release. If there's a positive for RDTs, okay, so they're immediately given an anti-malaria drugs, and then they send them home, okay.
Yes.
Right. So these are two different management, Malaysia
Mm.
and Indonesia. So from there, and even in Malaysia, even in Indonesia, in hospital, one of the hospital in Putu Sibau, I think it's Putu, yeah, we did found that one positive under the microscopy. So the microscopists in that hospital in Putu Sibau report it as Plasmodium vivax. But when we look under the microscope, it's actually Plasmodium nosai.
Mm.
So it shows that people or the microscopists in that hospital probably are not aware about how does a nosai look like.
Mm.
Okay, so there's a different angles here we have to look at actually. So it's all about how do you actually want to catch every single patient, febrile patients,
Mm.
and then you examine it thoroughly, For example, like in Kapit divisions, when you have fever, the first thing first that we will examine is malaria, followed by dengue and others and so on, right? Because in Kapit divisions, zoonotic malaria is endemic. I would say is very endemic. So they put that as a priority for diagnosis in the laboratory, right? So if I were to change,
Mm.
have the opportunity to change in what's going on in actually West Kalimantan, for example, in Putu Sibau, the first thing first, the people come with fever because how it is close to Sarawak, I
Mm-hmm.
would suspect malaria first
Okay.
before other species. Then you can see whether or not you really have high cases of malaria, or actually, there is a gap between Indonesian Borneo and Malaysian Borneo. So actually, it's all go back to the management system.
Okay. Is it also useful that we use the PCR if we want to I mean like to make sure that these are knowledge and these are not for the febrile cases, then
Yeah.
we can just screen with the PCR, or just we follow with the microscope first and then rely on this result and then back to the PCR or something like that.
So not actually, you really need a PCR because even in capit division, in hospital, in the government hospital here, they don't perform PCR. They use microscopy, right,
Mm.
to report it. The reason why they're able to report nosai because that area is known as an endemic for nosai, and then all of the malaria, P. malariae cases or P. vivax or P. falciparum are all imported when we look at their travel history,
Mm-hmm.
right? So it for at least for a start, a microscopy is still, I mean, you can follow the gold standard microscopy. So the first thing first is to train microscopists. How does a P. nosai
Not.
look like, which is similar to P. malariae. And then usually PCR is like a supplementary, additional, because if you want to confirm the species, further confirm the species, then you can do PCR. But you know how expensive PCR can be, right? So not all hospital can do PCR. University, yes, because university, when you can have funding, we can do PCR. But for the hospital, or even for the puskesmas, for example, right? So PCR machine is not is not a thing
Yeah.
there,
Mm.
right? But microscopy is the cheapest, and trained microscopy is actually required in that area. Yeah.
Okay. Yeah, thank you, Paul. And then another questions, Paul. So, in my lab, we did the genetic diversity of the mosquito vectors, and then we found the Anopheles complexes. And then we thought that there is the allopatric species
Mm.
because there's like a geographical separations in that area. Is that also possible, the like a capacity factorial or capacity of the factors to transmit the mosquito? And then it is also different between the Malaysian Borneo and Indonesian's Borneo
Yeah.
because the terrain is also might be different or
Yeah.
something.
Yeah. See, the mosquito is very complicated, huh?
Yeah.
Because you have found some different species of mosquito, different groups of mosquitoes, the complex and everything. They plays roles on transmitting the malaria parasites. So I do believe when you when you catch more mosquito, you can incriminate more species that able to transmit the parasites. Just like
Mm.
we have seen in Sarawak here. Previously, we only found that Anopheles latens are the
Mm.
only mosquito. Now, when my PhD students who is in the levels writing stage right now, uh,
Mm.
he found more species, Anopheles donadi, and different kinds of groups that also carries Plasmodium nosai
Mm.
mosquito. So, uh, how is this why it is so different? You have to go back to actually could this mosquito has adapt to a new environment,
Yeah.
comes to deforestation and so on. And how do adapt in the environment that you can explore going to do
Mm.
genomic analysis and so on to see the adaptations and how different are they between the mosquitoes nowadays compared to mosquitoes 10 years ago. How are different are they, you know so yeah, this mosquitoes are very complex
Yeah.
studies, I would say.
Yeah.
Absolutely complex, but it actually it tells us how complex is the infections in human relate to this diversity of the mosquito itself, actually.
Okay.
Yeah.
Yeah. two questions more, Paul. This is
Sure.
more. And then one question is about the captive monkeys. I heard from Janet that in Malaysia, many people, many locals, use the captive monkey to pick up the coconuts.
Yeah.
And then, is there any recent proof that the captive monkeys are also infected with the Knowledge? As previously Janet said that there is no proof of the captive monkey carrying the Knowledge.
Yeah. So since her time until now, we have not done any analysis of the captive monkeys recently, right?
Mm,
But why captive monkeys are negative for nosai? I think it's because the factors are not there. The factors where that surrounding this captive monkey, captive monkey usually live with humans, very close.
exactly. Mm,
Yeah, the vectors are not there. You don't see Anopheles, at least in Sarawak. I'm not sure about other environment, but at least in Sarawak, where we sometimes when we visit
mm,
houses where they have monkeys, captive monkeys in the house. Last time, nowadays we are not allowed to do that anymore
mm,
because
mm.
they are protected animals. So we don't see any other mosquitoes, Anopheles mosquitoes coming nearby,
Okay.
except for Aedes and Culex.
Mhm.
Yeah.
Yeah.
So that's why I think there's a big reason why these captive monkeys are mostly negative. Yeah, they are parasites.
Makes sense. Yeah. Okay, agree. And this is the last questions from me, Paul. If my students want to learn about the genetic diversity or the population genetics and then the microsatellite analysis, is it possible my students to come to your university or to your lab, Paul?
Yes, yes, of course. Very welcome to come here in even to do the attachment, few months attachment. Sure, no problem.
Yeah.
Yeah.
Okay. Oh, yeah. Because in the university we have a programs. We have the small funding for the students, and then I really want the students in the Master of Epidemiology or my undergraduate student of epidemiology to learn the modern epidemiology. How then we connect the conventional risk factor analysis with the molecular stuff, some kind of like this. And then we really need someone who are uh, keen to teach about the modern epidemiology, especially for the population genetic study.
Yeah, no problem. I mean, I'm open to that.
Okay, thank you very much, Paul.
Yeah.
Yeah, that's it from me, and then I will return back to Nabila.
Okay. All right. Thank you so much to Dr. Fauzi and for Professor Paul for the comprehensive discussion. Okay. I think it looks like there is no question here, so we move on to our next session. So once again, thank you very much, Professor Paul Cliff Simon Davis, for delivering such a clear and eye-opening presentation and for the interactive discussion session that we have had before. And also, thank you everyone for your active participation. So give a round of applause for all of us. Okay, now, before we move on, I would like to present a certificate of appreciation to our wonderful speaker, Professor Paul. Okay, to the committee in charge. Okay, please. Okay, thank you so much. Okay, so Professor Paul, I kindly invite you to turn on your camera, yes, and give us your best pose so we can capture this certificate handover as documentation. Are you ready? Okay, on the count of three. 3, 2, 1, smile. 3, 2, 1, smile. Okay, last one. 3, 2, 1, smile. Okay. Thank you very much. Let's give both our speaker another round of a virtual applause.
Thank you very much. Thank you very much
Okay.
for
All right.
inviting me.
Before that, I want to remind the participants who haven't filled the attendance form yet, please, that the link will be shared by the committee in charge in the chat room because e-certificates will be shared based on your attendance. Okay. As we come to the close of today's online session, we would like to extend our deepest appreciation to everyone who made this event possible, and our sincere gratitude goes to Professor Paul Cliff Simon Davis for delivering such an enlightening session. And we are also grateful to all of the academic staff, participants, and our team who joined this meaningful session online. We hope today's lecture has enriched knowledge and deepened our understanding of critical issues in epidemiology, especially in the complexity of zoonotic disease transmission. Okay, so we have come to the end of today's event. We are looking forward to seeing you again in the next visiting professor lecture. I'm Nabila as the host for today. Thank you very much for joining us today. Wishing you have a pleasant day ahead. See you soon and goodbye. Thank you so much, everyone.
Thank you, Paul. Thank you very much. Hope to see you very soon in Semarang, in Malaysia.
Thank you for inviting me. Thank you very
No
much.
worries.
Thank you, Fauzi.
Bye-bye.
Bye.
Yeah.
Bye.
Bye.
Thank you very much, Professor.